So we're going from outer space to the deep sea. Our research in the symbiosis department focuses on chemosynthetic symbioses, and you see here that we study them from a wide range of approaches, really in a holistic attempt to understand how these symbioses function. And the way chemosynthesis functions is chemosynthetic bacteria. We already heard a little bit about them. They use energy sources such as sulfide or hydrogen or methane and carbon sources, and they use the energy when they're oxidized, these compounds, mainly through oxygen in these symbioses, and harness that energy to fix CO2 into biomass and live in symbiosis with animals that use them as nutrition and then eat them. Remarkably, although they were discovered in the deep sea in 3,000 meters water depth, we now know that these symbioses are widespread in many environments, including shallow water environments right off of many coastal environments. And they've evolved numerous times, these symbioses. You see a phylogenetic tree or phylogenomic tree here, where all the colored lineages are bacteria that have evolved chemosynthetic symbioses with hosts. They're often closely related to shallow water bacteria, and there's a wide range of bacteria, 15 different bacterial groups that have evolved these symbioses, and 2 protists and 9 animal groups. And what that tells us is when we see something happening repeatedly in evolution and convergence, evolution, that there's a strong selective advantage, both for the bacteria and the animals, in coming together in these associations. We work on the deep sea mussels that I'll be telling you more about in my talk today, but also on the shallow water hosts that you see here. And we use a wide range of methods to work on them because the microbes can't be cultivated, and it's extremely difficult to keep the hosts in the lab for longer time periods, not to mention getting them to reproduce. So we go from the full range of omics to lots of different imaging methods and incubations in our field work. So let me tell you about deep sea mussels and how they work. What you're looking at here is a micro CT image of a Bathymodiolus mussel, and the symbiotic organs are the gills. They contain the symbiotic bacteria. They're very greatly enlarged because they house so many bacteria. We estimate that an 8-centimeter large mussel houses about 10 to the 12th bacteria cells, and they only have 1 or 2 symbiotic species. This means examining their microbiomes is relatively easy because we're not dealing with a great diversity of species that, for example, make studying the human gut microbiome so challenging. And the symbiotic bacteria sit inside the host cells. What you're looking at is a fluorescent in situ hybridization image of a gill filament. And what you can see here in the red box is a single bacteriocyte, a gill cell, in which the symbiotic bacteria, sulfur oxidizers in yellow, and in magenta, the methane oxidizers, are sitting in this epithelial cell. And you can also see that not all cells are colonized. The host, the outer tissues that are full of cilia and mitochondria, the host doesn't allow the symbionts to colonize these. Only certain gill tissue cells are allowed to be colonized. And these are, as I emphasize, epithelial tissues, similar to intestinal epithelium. So when you think about a gut, then what you can think about in these gill tissues is more like an inside-out lining, with the gill lumen in the middle here, where you see the macrophages patrolling, or hemocytes patrolling the gill tissues, and this is the region in which the symbiotic bacteria sit, and they're eaten through phagolysis, lysosomal digestion. But today, I'd actually like to tell you about a parasite, not a symbiont. These are some of our newest results that we recently published and that we find quite intriguing, and I hope you do too. So theory would predict that when you have these types of dense communities of any type of animal, we know that whether it's bird flu or in agriculture, many, or when we keep animals, is that they're an oasis for parasites. And we find remarkably few parasites, but the one that we did find is quite intriguing. And the two people that did the work are Nicole Dublier, who is now the head of, at EMBL, the head of Mobile Lab Services, and Miguel Gonzalez Porras, who did a postdoc at the MPI for Biology in Tübingen, is now at the University of Malaga. So the parasite that we found, a gamma proteobacterium, I'll show you a tree in the next slide, a single pathogenic microbe, Endonucleobacter, infects the nuclei of the deep sea mussels, and it replicates. You can see here it extends, then it divides longitudinally, and then it starts to septate. And in a single nucleus, up to 80,000 bacteria then grow from the single bacterium that infected it. And this greatly balloons the volume of the nucleus up to 50-fold. And at the end of the infection cycle, then what happens is that the nucleus and the cell burst and the pathogens are released and the infection cycle begins again. And as I mentioned, they're related to shallow water bacteria. What you see here is a phylogenomic tree. Up here is Endonucleobacter. And it's intriguing that they're related to a shallow water symbiont called Endonucleobacter that we like to call the E. coli of the oceans. It's been found in quite high abundances, for example, in corals. And people, there's a nice paper that came out about Endonucleobacter, which is frenemies of the reef. It's unclear for the Endonucleobacter if they're actually pathogens or commensalists or symbiotic bacteria. For the pathogen that we're looking at here, the related one, we can say it's definitely pathogenic. So only very few bacteria are known to infect the nuclei of cells, of animal cells. And the reason for that is you're hijacking the command center of the cell, so that you really, if you want to multiply, that's not a good place to be. On the other hand, if you want to hide from the immune system of an animal cell, it can be a great place to hide. So how does the mussel protect itself? Number one, the pathogen can only infect symbiont-free cells. So there are a couple of reasons we think it can only infect these, but we do not see it infecting any of the cells that contain symbionts. And here you see again a fluorescent in situ hybridization image of the, in green and red, the sulfur oxidizing symbionts, and where the pathogen is, there are no symbiotic bacteria. So what Miguel did was use laser dissection microscopy to look at non-infected nuclei of the mussel, then at early, mid, and late infection stages of the mussel to get a better idea about their metabolism during the infection, using transcriptomics then to analyze expressed genes. And in contrast to what we had first assumed, that they might be eating the DNA or the RNA of the host, we found no genes that were encoded for gaining nutrition for chromatin or RNA or DNA. And that makes sense when you think about it, because you want to let the cell actually be intact for as long as possible. And we see that the host cells remain transcriptionally and metabolically active. What it does is it gains its nutrition, the pathogen, from host sugars, lipids, amino acids, and possibly mucin, which is mucus on the outside, which is interesting because it's not quite clear how it accesses this. But we have some hypotheses about that as well. And it's clear that when you do the calculations of 80,000 cells, that the host cell it infects can't provide enough nutrition on its own. The resources, the host sugars, lipids, amino acids are drawn from neighboring cells as well. We finally asked, how does the pathogen avoid apoptosis? Apoptosis is programmed cell death, and that's one of the first responses that animals have when they're infected by viruses or by bacteria, by pathogens, is that they go into programmed cell death. We were very surprised to find that Endonucleobacter expresses as many as 15 inhibitors of apoptosis. And the reason we were surprised is because these proteins, these genes, are only known from animals. And they're not known from any prokaryotes. They've only been found in a few animal viruses from insects where the viruses have acquired them through horizontal gene transfer. What inhibitors of apoptosis do is exactly as the name suggests. They inhibit apoptosis. It's closely regulated in animal cells because obviously they don't want to start this cascade unless they have to. And the inhibitors inhibit caspases, which initiate apoptosis from beginning the apoptosis. And so what we see is a real arms race between the pathogen and the host in initiating apoptosis. What you see here on the left side are the expression patterns or the expression processes that we saw of the inhibitors of apoptosis. Black means not expressed. Only 3 are expressed in the beginning stages. Caspases, when it gets red, are not highly expressed. And when we get to the late infection stages, all 7 inhibitors of apoptosis here are being expressed. And you can see the host is ramping up its caspases to try to counter this. Finally, we were interested in, well, if this is an animal protein, where did the pathogens get it from? And in a phylogenetic tree of the inhibitors of apoptosis, what we can see is that those of the pathogen are most closely related to those of its muscle host. So you see here in light blue all the mollusks, that bivalves and other organisms that have the inhibitors, and you can see that in dark blue the pathogen inhibitors are most closely related to those from their muscle host. Intriguingly, we have a Russian doll system here and evidence for the fact that the pathogen can be infected by viruses. Here in a TEM image, you see the nucleus, you see the bacteria inside the nucleus, and you can see capsids that are typical of DNA viruses that are infecting the pathogen within the nucleus. And so our hypothesis is that the pathogen acquired these inhibitors from their muscle host. That's clear from the tree. This is surprising because horizontal gene transfer from eukaryotes to bacteria is rare. Eukaryotes, as a rule, don't have a lot to offer in terms of their metabolism to bacteria. It's more the other way around. But what eukaryotes can offer to pathogens is ways to protect themselves against their immune systems. And we are hypothesizing that viruses might be the mediators of this horizontal gene transfer between bacteria and eukaryotes. And as is known also from the human gut microbiome and other environments like biofilms, physical proximity of microorganisms promotes horizontal gene transfer. And that's certainly something that we're seeing here. And with that, I'll finish up by highlighting the fantastic work that my group has been doing and the many sources of funding that we have. And thank you for your attention.